My name is Dave Smith. I'm with Lonza. I see there's maturing nature occurring in the cell therapy world. Just a few years ago, we were happy with our first products being approved. Wound healing products, orthopedic products. We're treating thousands of patients with products now, up to 10,000. But the maturing process is occurring. What's going on now is we have our first products in phase 3, which are going to treat not tens of thousands, but hundreds of thousands of patients, which causes great logistical changes and great challenges for everyone involved. A few years ago, you could do a clinical trial, 100 patients. Now you're talking about multinational, thousands of patient trials. We need to, and with that, we also have to look at our business model. Pharma is now becoming involved. They're expecting much more when they're talking to us. They're expecting us to know how logistics is going to occur, how we're going to make universal product so that one product they make in a facility in the US can be used in every single clinical trial and every single patient across the world so they can transfer it back and forth. They're expecting us to understand our funding. They're expecting all of these things, this maturing of the market, where much more is expected of you out there. And here is a chance for you to talk to this distinguished panel and have them answer some of the questions that you have today. As an introduction, if the group can take a minute.
Sure, I'm Dr. Gil Van Bockelen. I'm Chairman and CEO at Athersys. That's my day job. Had a chance to talk a little bit about my background yesterday, but I can briefly summarize what we're doing at Athersys and I think why that makes it relevant here. So we're focused on developing a product called MultiStem, which is an off-the-shelf allogeneic stem cell therapy that has a pretty distinctive profile. So first off, full disclaimer, I'm a big believer in all forms of regenerative medicine, autologous, allogeneic, tissue engineering, even the non-cellular based forms of regenerative medicine, which I think have transformational potential as well. Our focus at Athersys is really on a scalable allogeneic product that we think has some distinctive properties and has shown promise across a range of different therapeutic areas, which I'll cover during the course of the discussion today. But Bob Deans, who's our Executive Vice President, or affectionately known as the Grand Poobah of Regenerative Medicine at Athersys, is actually going to be giving a more in-depth discussion a little bit later today to talk about some of the ongoing clinical work we're doing. But we think a lot about the logistical issues that you just described, and certainly anxious to talk about that.
Hi, I'm Eduardo Go. I'm the CEO of Tygenix, a Belgian company, and we have an autologous product, actually the only cell therapy product that is approved and launched in Europe, and we have a platform of allogeneic adipose-derived stem cells, which is currently in phase 3 in a non-very glamorous disease, complex perianal fistula in Crohn patients, and we also have finalized a phase 2 in RA.
Sam Cava. I'm Vice President of Neuroscience at MedPace, full-service CRO. Before joining the pharmaceutical industry, my background, I'm a neurologist by training, and I was in academia. My focus was neuroimmunology, neuro-oncology. I was part of the cutting-edge therapies at that time in neuro-oncology when biologicals instead of chemotherapies and vaccines and monoclonal antibodies were starting. And now it's certainly exciting to be part of this new wave of therapeutics. Like Gil, I'm a great believer of regenerative medicine, especially in areas where other modalities clearly failed or the diseases are too complex to target them with more targeted therapeutics. And I'm happy to be here today to share some of our experience in cell therapeutic trials that we've done over time.
Tim.
I'm Tim Fong. I'm Vice President of Technology and Process Development and Product Development at PCT Cell Therapy Services. PCT is a cell therapy development and manufacturing organization. We have broad experience over the last 13 years in developing many different types of cell therapies, ranging from straightforward CD34 selections to very complex types of manufacturing for cell therapeutic processes. And very excited to be here. We bring a wealth of experience in dealing with a number of different clients, all the way from basic research, helping them develop their process, to a commercial process for manufacturing.
Thanks, Tim. So the first question, Eduardo and Gil. If you've now fairly advanced in your clinical trials, however, everyone has done those things now that you said, oh, if I had only known back in phase 1, I would have done this. So if you could have turned back the clock, what are the things you would change now and redo and do it a different way? I think it will help the group.
Yeah. As I think back operationally in terms of how we've approached things clinically, honestly, I'm not sure that I would change much there. I think we selected a bit of an unorthodox approach in terms of using a broad distributed network of collaborations to do a lot of the preclinical work. I think that was absolutely the right move. If you're a small company, it's very difficult for you to try and do it all internally. It's just impossible logistically, and it would be way too expensive. So we've really relied heavily on our collaborative relationships with dozens of investigators from over 30 institutions across the US and in Europe. I wouldn't change that. In terms of our approach with the FDA, I guess one piece of guidance that I would give people is don't come at the FDA with an adversarial mindset. You really should come at them with a collaborative, collegial mindset, or at least that would be my strong recommendation. I think you'll get much more out of the dynamic that way. We did something a little bit different than a lot of companies do when they're moving programs into clinical development. Instead of asking the question, what's the least amount we have to do to get this program into clinical development, we actually told the FDA we wanted to have a series of formal sit-down meetings with them over time. I think most of you know that normally you only get one pre-IND meeting with the FDA. We actually had 5, but we were able to have 5 because we had a product platform that had relevance across a range of different disease areas. And we told the FDA that we wanted to use that series of discussions as an opportunity to really answer all of their questions about safety, looking at things like biodistribution, product characterization, and a whole bunch of other things. And I think that was something that we did that I think has actually worked quite well because it established a strong rapport for us. In terms of really painful lessons learned or things that I would do a little bit differently, it typically revolves around money. When we became a public company back in 2007, interestingly enough, most of the institutions couldn't care less about our regenerative medicine platform or what we were doing with multi-stem. They were really interested in our pharmaceutical programs. But nonetheless, there were people that saw that had promise, and so we had a lot more demand for the financing. Our goal was to raise $30 to $40 million, and we had $150 million in institutional orders. And I wanted to take as much money as possible. Some of the key institutions said, no, we only are willing to go so far. So I guess one lesson is, and it's an old banker's adage, that whenever the money's showing, take it, right? Take as much as you possibly can. We probably could have taken $100 million in that financing. We ended up taking $65 million. In retrospect, I wish I'd fought a little bit harder. And in fact, the thing I will never forget saying to the lead institutions was, the world could change in a heartbeat. A year from now, this was in 2007, a year from now, the world might be a very different place, okay? And they said, no, that's not going to happen. Everything's fine. And, you guys know the rest of the story. So I would say, for those of you that are thinking about doing fundraising, fight hard for every nickel, spend it very carefully, because you don't know how long it's going to last, and really stick to your guns. If you know you're doing the right thing, then just fight hard and don't give up.
And have Gil manage your portfolio.
Yeah, well, yeah.
He's got wisdom there.
Be careful what you wish for there. Yeah. So,
Eduardo.
How long do we have? How many things can we change? No, if I could do 2 things really differently. One, looking at the new company and Condor Select would be, I think that the company really underestimated the effort, work, and resources needed to get the product reimbursed. And I keep telling the people that we've been pioneers having those discussions, and as the first pioneers that went west here in the U.S., sometimes you're killed by the Indians. So most of the times we're surviving barely, and it's taken. The product was approved in Europe, just to give people an idea. The product was approved in October 2009. We got first reimbursement in May 2011. So that's a year and a half telling your investors that you're going to finally you're going to make some sales, and then it takes one small country, that's Belgium, 10 million people. Second country came June 2012. So we're hoping to get now Spain and France by the end of this year, beginning of next year. But that shows that is 3 years from approval to actually getting some reasonable sales. So it was a very painful, and I think that we didn't realize at that time how tough it was going to be and how much work you need to do to get enough data to convince the payers that they should reimburse a compound. And looking at our platform, I think that it's funny to try to change things a few years later. It's always very easy to guess what you should have done differently, but we probably should have moved from our autologous product to our allogeneic product earlier on, but that's as I said, very easy to say now. And also looking back, probably our first phase 3 with the autologous program, we rushed a little bit into an indication that we were a little bit less certain, and we paid the price because it was not the right indication. We couldn't find the difference that we were looking for. So again, when you're small and you don't have all the money that you want, you tend to cut a lot of corners and you try to go a little bit quicker than what you should go. I think that it pays off to go step by step and really making sure that every time you build into something that you're certain. Otherwise, going back to square one when you're middle of the way, it's a pretty painful exercise.
Would you put pharmacoeconomic data parameters in your clinical trials, more of them? Would that have helped you on the reimbursement?
Yeah, but it's not only getting the pharmacoeconomic data on the clinical trials, it's preparing a dossier and making sure that you have data on the cost of the disease and the available treatments and how much payers pay. And let's face it, and it was discussed yesterday. I thought that the panel was very good. We're going to be facing an issue in which we're going to ask payers to pay a lot of money upfront to avoid some of the long-term costs of some of these diseases. And it's not an easy discussion, and sometimes you are faced with the decision, well, show us the data that in 30 years' time you're not going to have osteoarthritis. Yeah. Well, I can wait 30 years, but even by then most of my patients will be dead, so what's the point? Yeah. So, it's funny how for some of these diseases, I always get the comparison of the, papilloma vaccine. There's no proof today that the vaccine will treat ovarian cancer in the future, will reduce. There's a very good idea that it will be the case. So, we're vaccinating all the 14-year-old girls in the world, which I think that makes a lot of sense. Why do we have to really prove that indeed, by, curing the cartilage, you will not have osteoarthritis in 30 years from now? I think that there's enough rationale, and you need to convince the payers that it makes sense. But it's an uphill battle when you are, a 30 people or 40 people organization.
Yeah, I think there's actually two important lessons there. One, most people, I think, particularly from smaller companies, they think about preparing for the reimbursement issue as they're approaching the finish line, right? That's way too late. You need to be thinking about what you need to do on the reimbursement side frankly, as you're getting out of phase 1 and as you're moving into phase 2. And frankly, even if you're thinking about it in phase 1, that's a good place to be thinking about it. I think the other point that Eduardo just made is there's a wealth of tools that are now available, thanks to some of the companies that are here this week and others, that are giving us early, powerful indicators, biomarkers, and other things that even if we don't want to run a 10, 15-year study, because that would make it totally impractical. By the time we get to the end of our IP, by the time we prove the point, right? We can't do that. But there are powerful ways that are emerging that allow us to create a very compelling picture for the third-party payers, but we need to be having that conversation earlier in order to really get their kind of buy-in and educate them, because they really, frankly, most of them, they're only going to think about us when we get in front of them and say, you need to be thinking about us. And when I say us, I mean collectively the industry. So.
And one final thing, in Europe, you're facing at least 17 reimbursement authorities. So
yeah.
The reimbursement dossiers are different in each country, and the arguments are different, and the approach is different. So.
Great. Sam and Tim, you see as suppliers, service providers for the regenerative medicine market, you see a large number of companies. What's the gaps, or what wisdom should you be giving to these the groups here, looking at across all the companies that you're working with?
I'll start. Actually, there are few, but let's take one to start with, and that will build on what Eduardo said. It depends really on the type of cells. That the problem, of course, that comes from the type of cells are different, but there's no question that autologous, everybody knows that, that's going to be practically very hard. And not only commercialization-wise, but even running the clinical trials. Because every time you're asking the sites to do things that are outside of the standard of care of a given disease, then you're getting into more trouble and more cost than you need to. So that's simple. So I know that in lots of cases, there's no way around that, and that's your platform. But of course, there has been some examples where you can shift your process a little bit and explore different alternatives. The other thing, of course, that is very related to that is the route of administration, or how you administer your treatment. That also will have lots of implication on the practicality of your trials, which is an important step in commercializing, and the cost of these trials. It's not easy to find sites who can do all the procedures that are needed for a given route of administration, and at the same time have the appropriate number of patients to enroll on the study. So you end up investing in many sites for each of them to enroll 1-2 patients, which is very not economic in terms of running trials. And when you go into larger trials, phase 3 studies, it's not only the cost, but then running out of possible sites that can really do this kind of thing. So take an example of a stroke study. You're using autologous stem cells, and they have to be delivered intraarterially. You already see the challenges, right? You need now, when the care of a stroke patient is usually done by a neurologist, now you need a hematologist to do your bone marrow aspiration. And then you need, of course, to send the cells and get them back, but then you need an interventional radiologist or neurologist to inject them in the artery. So it complicates matters a lot, and then you have all the cost that is associated with it, and how many sites would be able and willing to go through all that coordination effort. And another big challenge, of course, is, again, the degree of processing that your cells need at site, at the point of treatment. And the less processing you need, that, of course, the better. It sounds like very clear, and Gil, I guess, mentioned their platform has that advantage. But you can, of course, train sites. You can make them part of your manufacturing process, so to speak, and you can make sure that there's enough quality control. But it's not always an easy task. It requires lots of extra steps, lots of resources on behalf of the manufacturer, the sponsor, and the CRO involved. So if there's any way that you can simplify that and get them a ready product, that, of course, it would be worth the effort done ahead in preclinical and in the manufacturing phase to simplify these, so you don't have to encounter all the costs and the complexities down the line when you get to clinical development.
Thanks, Sam. Tim?
Yeah, for us, we see, and maybe to build on Sam's comment, is that one of the things that we see when clients come to us is that they haven't thought about cost of goods. And for many of our clients, particularly the ones in early stages of development, a lot of the processes and cell products and ideas come out of an academic or university environment. And so for the early clinical trials that are in which their product is manufactured in a university setting, the cost structure there is different than in commercial manufacturing. And it's not only the labor, but it's also facilities and sort of the regulatory and quality systems put around these processes. So, coming into the phases where you really think about, instead of making a dozen or 2 dozen products per year and scaling up into 100 or maybe 1,000 a year or maybe even 10,000 a year, I think that the processes that are brought in from the academic institution into a company such as PCT, we certainly can, quote unquote, GMP it, make it more GMP compatible, close up the systems, maybe do some semi-automation in the whole process, but fundamentally it's the same process that's brought out of the university or that was developed in the university. So, and again, these types of costs associated with making products at this level are reasonably acceptable for small clinical trials. Again, a dozen patients or so, or maybe 50 patients over a year or 2 years. But once you begin thinking about true commercial manufacturing, 10,000 products per year, a lot of these processes and the structure around it to support the manufacturing just is not financially viable. I'm sure all the panelists have looked at this and thinking about, well, it's okay to make 100, but now we're going to make 10,000, and you simply can't just multiply your cost by 1,000. That just won't work for your models.
Yeah.
And certainly that has a significant impact on your reimbursement strategy.
Yeah, I think for people that are developing allo products, so I think about our stroke program. The need there is kind of mind-boggling. You think about 10,000 patients a year. Well, in the U.S. alone, there's about 800,000 patients a year that suffer a stroke, and about 85 to 90% of those are ischemic strokes. And guess what? Only about 5% of them, give or take, are getting TPA. So you're talking 95% of the patients out there that are basically getting the clinical equivalent of hand-holding. Now, if you want to be able to penetrate that kind of an opportunity, if you look at the U.S., Europe, and Japan combined, you're talking about 2 million patients a year, right? So you've got to think on a scale that's fundamentally different than I think most of us have historically really been kind of approaching it. So I think your points are exactly right. And not that Eduardo and I wouldn't be happy with doing things that involve tens of thousands of patients per year, but I think we've even got to think well beyond that. And so you really have to be thinking about the economics of your production system, and that's where I can speak from experience, because we work with you, we work with you, we work with you. We're not working together yet, but I'm sure we can. We had a good time on the golf course, and I'm sure we can figure something out. So it's I think you really do have to be thinking about the scale, and you got to be thinking about it early on. It's doing something at the benchtop level is so fundamentally different from taking that into an actual production environment where you've got to be worried about GMP, and you've got to start. If you don't know what your cost of goods are, you've got an issue, right? I mean, you may have a great cost of goods profile, but a lot of people that aren't really thinking about that or where they need to be in order to make it commercially viable. And I think, frankly, that's a common malady in the field. People aren't paying enough attention to that.
And there's no easy solution or simple solution.
Right.
It's, as you think about true commercial scale manufacturing, you think about automating your system so that you have less manual labor. Perhaps you use specific devices to grow up, scale up, or scale out your process. But it's a balance of when do you invest in that automation? For early companies, to produce half a dozen or 24 lots per year certainly doesn't make sense to invest early on. But at some point in time, as you begin to move through phase 2, phase 3, and ultimately think about commercial, you've got to make that decision of when do we start really investing in commercializing this process.
Well, and just to put one more point, you have to lock down your manufacturing approach by the time you're going into a registrational study. So if you start thinking about solving for that problem when you're lining up your registrational study, it's way too late. I mean, you've really got to be thinking about it at the beginning of phase 2, about how you don't necessarily have to have it solved then, but you've got to be thinking about how am I going to get there by the time I'm ready to move into a registrational study, particularly if you're going after larger indication areas.
And I think, just if I can make a comment, something that has changed significantly a few years ago when we were having these discussions, I think that the enabling technologies were really behind. I think that the advantage today of the companies that are reaching these stages of finalizing phase 1 is that there are some proven and available automation systems, which were not the case a few years ago. So again, if I had the opportunity of going back, probably we would have done more in the past. I don't think that it was the right thing to do then, but I will make sure that if I was today at the stage I was a few years ago, that I would incorporate them into my equation.
So, two points. One is, Tim, the other thing I think you and I deal with, and I think Rob, is that if your cells aren't well characterized, then you have the other issue, which is when you change the manufacturing process, do you really know you have the exact same cells?
That's right.
So the cell characterization and doing that early and not waiting till, I only have to do it in phase 3, if the registration, gets to be very challenging for the people who are doing process development.
And risky for your program, because you don't know if you can duplicate the results unless you have that consistency.
That's right.
Second, I hear everyone talking here, lots of things to work on. There's a lot of service groups out there, there's a lot of knowledge out there. Where are the gaps today? The things that are going to kind of jump up and hit these companies as they're moving to phase 3?
I'd say one of the biggest, well, there's gaps on so many different fronts. I'd say one of the biggest things is making that transition from the modest-sized production systems into the truly, for those people that are focused on that, right? If you're an autologous business model, and again, I'm a believer in that, I think that's going to have real relevance in certain areas. But if you're an allogeneic product, you've got to think about How am I going to go from something where I can produce dozens or a few hundred doses at a time to producing tens of thousands to hundreds of thousands, large quantity, consistently, right? In a very well-validated way, which the FDA is going to be comfortable with. And I think that there's lots of exciting things that are happening on the bioreactor front or other technologies or the integration of various technologies. But frankly, for a lot of us out there, that stuff can't happen soon enough, right? It's got to happen sooner rather than later. You don't want that to be the ultimate impediment that's holding you back. Everything else is working, and yet you've got to take extra time to kind of solve for that stuff. So in my mind, I think that's the biggest challenge and opportunity for a lot of people in the field, is crossing that bridge of scalability. Because that's when the technology, the power of the technologies really gets unleashed, right? So in my mind, I think that as we're getting there and we're demonstrating clinical proof of concept through robustly designed, well-validated studies, which is another thing we really need to make sure we focus on, that those things in concert will be a big part of the solution.
One comment there, I think that it's not only that the technologies need to provide us the scalability, either it need to provide the cost reduction thanks to the scalability.
Exactly.
So, I think that today, at least us, we're facing that as we go from 100 to 1,000. Great, we have a better system, but at the end of cost of goods doesn't change, and we go to 5,000, and we seem to be making no improvement. I want to see when we go to 5 million, can we really produce cells at a fraction of the production cost today? And I think that would be the big step forward, because I think that we're going to be needing a lot of cells. I mean, if we go to some of these indications, I mean, you were mentioning stroke, we're working on RA. I mean, if you want to dose maybe every 3 months, 100 million to every RA patient, we need a lot of cells.
Yeah.
And we need them cheap.
Yeah, I completely agree. I think there are a lot of device manufacturers now who have targeted and making very significant strides in developing automated devices for expanding cells. But one technical area which people seem to forget is that you can grow up tens of millions of cells easily now, relatively easily now, in a number of different devices. But how do you fill and finish? Yeah. Right? You're not going to take your 10 to the 10 cells and have 4 people manually pipetting them back into vials. So again, I know that there are device companies that are beginning to think about that more on a pharmaceutical scale. How do you vial up 10,000 vials of Herceptin, for example?
And validate that vial 1 is equal to vial 100,000 and so on.
Exactly, and it's a little bit more complicated because with cells, technical things like, well, they settle. Well, antibodies aren't really going to settle over a span of an hour, but cells will.
Yeah.
So again, I think that device developers, particularly in the pharmaceutical field, are beginning to think about things like this. And these are, I think, some of the technical gaps that, as an industry, we really need to start focusing on.
And we would add to that, with the fill-finish line, on a pharmaceutical line, the standard line is designed to run 24 hours a day, 24 hours to get their long fill lines. This, you have 1 hour, maybe 2 hours at max to fill all the cells, and you're going to lose cells. So these lines have to run very fast for a very short period of time. And so it's a different concept, but you're right, that's one of the big areas.
But I think you should. I mean, some of the companies that are on this should really draw the parallelism with monoclonal antibodies, which today, everyone talks about, like if it was. Of course, everyone knows about them. But, when the monoclonal antibodies were started to be produced, and I'm too young to know, but it was those 5-liter bioreactors, then 50 liters bioreactors. Now it's. I don't know what's the size of the bioreactors, but at the beginning there was a lot of skepticism that the monoclonal antibodies could be produced in sufficient quantities at lower enough cost. Well, the companies that miss the monoclonal antibodies miss the market.
There's one interesting complexity there, though. So we talk about antibodies all the time as being kind of the precedent, if you will, or the case study that in many ways I think really shows us the path for cell therapy. But there's one big difference. So with antibodies, with secreted proteins or antibodies or anything like that, what you're doing is you're growing up lots and lots of cells. They're secreting lots and lots of antibody or lots and lots of protein. You're harvesting the supernate and you're purifying the protein and you're throwing everything else away. Okay, now what are we doing? Well, now we're growing up lots and lots of cells. We're throwing away all the easily processable stuff, the liquid stuff, and now we're left with the cells, right? And how do we get that out of the 2,000-liter bioreactor in a way that's rapid, efficient, consistent, or whatever it might be, whatever production system. So there's technologies which you can speak to at length.
Well, the other interesting one that comes out of it is you get to a point where, because of product doublings, your bioreactor hits the ceiling very quickly because you used to do, you did your study, you did it up to 25 or 35 PDLs over 8 passages or 9 passages for your.
Yeah.
That's a single bioreactor run. It almost comes down to 1 master cell bank per bioreactor, and that with the media that's involved, we've done estimates that it's.
Are we going to talk about the FPS problem?
Yeah, to run a single bioreactor, a 500-liter bioreactor, 1 master cell bank, it's about $5 million because of all the media and that goes into it. So
Right.
you're never going to go that big. There's going to be a cap in this just for, I can't afford to screw up that big.
Yeah.
So let's assume you have all this ironed out and you have a very good economic way to get your cells, then naturally these expensive novel therapies are going to go in areas of unmet medical needs. And guess why these areas are still unmet? Because they are very hard to treat and because lots of clinical trials have failed. So that's where another reality check, that lots of these sometimes smaller biotech companies who are developing very innovative, great techniques, but they go to the clinic and now they are shocked with the size of these clinical trials that would be required, the number of patients that are needed, the softness of endpoints that they have to be collected on these trials, and sometimes the length of therapy. So that's where it becomes also tricky. So if you're trying to tackle something like Alzheimer's disease or stroke and stuff, you start seeing these challenges that.
Although one of the advantages we have, I think, I'm not absolutely convinced that we're all going to have to run multi-thousand patient clinical trials for some of these things. I think it depends on how big is your delta, right? So how big is your effect, and statistically how many patients do you actually have to have in your study to be able to show that? And I think that for some of the biggest areas of unmet medical need where the current standard of care is either non-existent or for all intents and purposes is non-existent, you might be able to get there with, particularly with some of the recent changes of thinking at the FDA and other regulatory agencies where they are creating a path that will enable accelerated approval or breakthrough therapies, right? Absolutely. But what you absolutely must do is you have to show it in a properly controlled, robust, well-defined within the parameters of what the FDA wants to see, double-blind placebo-controlled study that's statistically powered, that includes dose ranging, right? Because the FDA always wants to see that. That's how you get to where you want to be. It may not be the case that you actually have to run a 3,000 patient study or something beyond that. But you've got to take a look at it on an indication by indication basis. And my counsel would be don't go for the studies where you think that the delta is going to be small and therefore to statistically get to where you want to be, you're going to have to run.
That's right. It depends what you're trying to do. So if you're after like disease modifying indication type thing, then it's inevitable that you'll have to run long trials later down the line. But if it's not necessarily that, just you're looking for incremental improvement in therapy, then absolutely correct. And that taking advantage of these regulatory facilitation techniques, so to speak, the fast track and similar things, you may get away with using surrogate markers if they are, of course, validated for that indication and stuff, and arguing that, okay, reducing the size of that stroke may be enough to say that the patient will do better 3 years down the line.
So I'll add one more to the list, which I think is going to come up strong, and there's not an answer right now, which is you're doing your stroke trial. You're treating these patients. How do I deliver a cryopreserved product to City of Hope and to Youngstown, Ohio, and to someplace in the middle of Maine? And I'm a hospital pharmacist. Hospital pharmacies aren't ready to receive a cell therapy product. If you don't have a blood center, how are you going to deal with these logistics?
Yeah. Well, I think we're going to have to actually educate the pharmacies much the same way that they had to be educated through the use of other types of biologics, right, where they didn't have that infrastructure to start with. But when they sense the need and people in industry actually educating them, I personally think that it's got to be regional distribution points, either the clinical centers themselves, where you've got secure pharmacy distribution capabilities for an allogeneic product, right? Particularly for acute indications where you want it, time is of the essence. You want to get it there as quickly as possible. And I think you want to do it ultimately, the ideal, okay, this isn't the only way to do it. And again, I'm not arguing against autologous-based approaches because I think that particularly in areas of unmet medical need, they can be hugely important and transformational. But for allogeneic products, I think the ideal is literally something that is as close to off the shelf as you can be. Something that's in a vial or in a cryo bag. It doesn't require any processing. It's controlled rate thaw, right? And you can get it right from when the doctor says, I need a unit of whatever it may be, that the pharmacy can deliver it in a very short period of time without having to go through a bone marrow type processing facility. That's the simplest path. Now, it may take us a while to get there, but I think that if we are able to deliver those types of easy, practical solutions, then I think that's where we're actually going to have the greatest amount of impact.
Now, if you're locked into an autologous type approach, then I guess the challenge is to develop that streamlined way to do it efficiently and fast throughout your sites and ultimately in your point of use.
Right, and for things like stroke or for other indications, if your window's big enough, right, then congestive heart failure or whatever it may be, right? I mean, you can pick your indication that you're interested in. As long as you can do that efficiently and do it in a way that is consistent and maybe routinized, maybe that's not the right word, then those types of therapies can be equally impactful and effective. Again, I'm a little bit biased. I think that ultimately, if you make it as off the shelf as possible, that will ultimately have a bigger impact because clinicians don't want to have to think about it. They just want to have to be able. They want to be able to do it in the shortest way possible. They don't want to learn. If you talk to interventional cardiologists or others, they don't want to learn a whole new skill set in terms of being able to do the things that they want to do. Right? They want to diagnose the problem, figure out what the most effective therapy is, get it into the patient as quickly as possible. So I think our collective objective is to get there as quickly as possible.
Great. Other comments before we turn it out to the audience? Sorry, I got to find some place where the light's not quite in the center. Questions for the group? Or Bob? Sorry, somebody has a microphone. I'll give you a little headset.
Following up on the last bit, are we, in listening to what you were saying, Gil, is the time now to start dealing with these issues about the transportation and processing in the hospital? I mean, there are some pretty substantial clinical trials that are starting, and I was just at AABB 3 weeks ago, and it's a big topic of discussion. It was almost like it came as Sandy did this weekend onto the blood banks and the hospitals, that they have to now have a process for dealing with this, and it created a tremendous amount of turmoil in a couple of institutions. I keep hearing in the future, but is the future now? Do we need to start dealing with this issue of what are we turning over to the practice of medicine now and how it's going to be handled in the hospital or clinic?
Well, so it's interesting. So I was actually at AABB as well. They asked me to come in and give a talk about this because they're really anxious to learn about it. I think that's a classic example where there is tremendous opportunity for complementarity and synergy between what they're doing now and what they see as the future of their world and what we're trying to do. Okay, if we try and build it from scratch, it's going to take a lot longer, it's going to be a lot more expensive. If we can find other folks out there that see this and recognize it for what it is and are willing to basically turn their ship in the right direction so that our interests are aligned and we can actually take advantage of some of their infrastructure expertise and other areas of commonality, then I think that's going to benefit everybody, right? Because they see their business changing in a pretty profound way. So they're either going to get swept aside in some ways, or they're going to be part of this, bold new vision of the future of medicine. But to your question, absolutely, I think the time is right now. That doesn't mean we have to do it all in the next 12 months, but I think we need to be thinking about these issues. And frankly, there's one other thing that I think is really beneficial here. So I always we don't have a relationship with Genzyme, but I always point to people like Leslie Wolf and some of the pioneers that have been working at Genzyme for a long period of time that started thinking about these logistical issues 20 years ago when Genzyme was first kind of breaking through into cell therapy and figuring out how are we going to get something from point A to point B and then back to point A when we need to. And they were thinking about the logistics of shipping and constant temperature environments and how do you monitor that and validate that. I mean, we owe them a big debt of gratitude collectively, I think the industry, because they were doing some of the hard early work. Well, we can learn from that kind of stuff. And I think as long as we're willing to communicate some of that knowledge to each other and share some of it, I'm not saying every aspect of what we do, but I think that that's going to benefit the entire field. And frankly, that's what the Alliance is really about, is trying to promote common awareness about some of these common challenges and how we can develop practical solutions for them. But the time is now, in my estimation.
Bob, I also talked, I'm a hospital pharmacist by training. I talked to the American Society of Hospital Pharmacy group. They're shocked. They're like, oh, we just started talking about biosimilars. That they're behind, they're looking for help, they're looking for wisdom, but they have no idea, realistically, when I started talking to them, they had no idea this was even on the horizon.
Yeah. And they won't until we get out there and talk to them and say, this is coming, okay? You can be part of it. We can work with you to figure out practical ways to address this, but absolutely, we got to start that process now.
And the odds are whatever you use in your large phase 3 studies, the same process has to be carried through your commercial use.
Yeah.
As long as AABB was mentioned, I thought I better pipe up, since I'm the director of the Center for Cell Therapies at AABB.
And we're so glad it's you, by the way.
And I just want to remind you not to abandon blood centers, because we have a 65-year history of doing cell therapy under quality systems regulated by AABB. And there are many, many blood centers now who are the core of the new cell therapies that are developing in their institution. So you don't have to rely necessarily on the hospital pharmacy. And our center is in the process of preparing the blood centers. We have 2,000 center members for this new field.
Good comment. Thank you. Another question?
Hi there. Quick question. I was hoping you might dig a little bit deeper, maybe Gil and Eduardo, on the methods you use to really convince payers. Because I'm curious about that dialogue for one very important reason, which is I used to spend a lot of time talking to payers, and they basically said, every commercial payer I ever spoke to said, don't waste your money on pharmacoeconomic studies. Because first of all, you're doing the study, so we don't believe the data anyways. And secondly, and perhaps more importantly, as a commercial payer, I only hold a life for an average of 2 to 3 years. So you're asking me to pay now for a benefit in 15 to 20 years. It's not going to accrue to my book of business anyways. So I'm curious, how you had the conversation and to what degree it differed for government payers versus integrated payer provider networks versus guys who have a big ASO book of business.
Yeah, you start, Noel.
Dialogue is a very nice word. I think that if you don't have the pharmacoeconomic data, that will be the easy excuse. So you need to have the data. I'm not sure that it is what they, at the end, will convince them. Funnily enough, and I fully agree, we had a better success rate with some of the private insurance companies in Europe. So for example, in the UK, both Bupa and Aviva, two big insurance companies, have agreed to routinely reimburse Control Select, while still the NHS has not agreed to the same, which for me is a very surprisingly effort. And exactly the same in Spain, we're selling a few patients through private insurance companies, and still the Social Security has not agreed to reimburse the product. So I fully agree, but it's like, private insurance companies seems to understand this idea of, it's better to spend a little bit extra money today rather than keep paying forever. I have to say that on cartilage repair, the argument is also less clear. I mean, for us, it's a pity that it's not a more, you're not talking about saving lives, because the fact that you're going to avoid someone being in a wheelchair 15 years from now, some of the maybe providers do not care that much. But unfortunately, that's the problem we have. So those are the arguments that we need to use. But I think that if you have enough knowledge of what is the real cost to society of those patients, that's the best you can do. And after that, you know, it's also a political decision. For us, a big thing is total budgetary impact. So, you know, the face price, you know, on a per patient basis looks extremely high, but we treat very few patients. So at the end, the budgetary impact in Spain, and I've made the calculation, we've spent more money from the government already discussing the price than if they had agreed to reimburse every single patient from day one. But unfortunately, that's the system. Now, for large indications, that will change significantly. So I'm sorry I didn't help a lot, but that's as much as we can.
So just to add a couple of thoughts to that. So third-party payers will tell you, we actually heard it from Stephen yesterday, right? And he's an ally, right? He's a friend and an ally. He wants to see this new world of medicine become a reality. Third-party payers, whether you're talking to CMS or whether you're talking to a for-profit or a non-profit or whoever it may be, they will always say, oh, we don't take cost into consideration. Okay. But you also, and this is why I asked the question yesterday of Stephen, hey, if we have a therapy for treating some significant area of unmet medical need, and we can show that within some several-year time frame that we're reducing in a meaningful way hospitalization or treatment-related costs or whatever it may be, okay, so that you can show the lines actually cross, are you going to be excited about that? So you're improving the clinical outcome, you're enhancing patient quality of life, and you're shifting the cost curve in the right direction. I can tell you that falls into the no-brainer category for them, okay? And every third-party payer you talk to, and I've had this conversation with a few of them, that's exactly how they look at it. Now, they may not explicitly in their decision-making process ever at any point along the way say, if it doesn't happen, then we're not going to approve it. But let's take a look at some of the recent examples of success, okay, the Dendreon issue, right? You get a really nice high reimbursement point, $90,000, but is the technology being embraced as fully as it could be if it was $20,000? Clearly it's not, right? That's not a pejorative statement on Dendreon. It's not a pejorative statement on anything that they've done. It's simply the practical reality of the fact that there are certain economics that will work and certain economics that will work much better, and there are some economics that just don't work at all. You know, if you develop a therapy for something like stroke and you can make the patient better 100% of the time, but it costs $1 million, do you think that's going to be practical? Of course not, right? So you've got to figure out where do we get into that no-brainer kind of territory, and you may be able to not get the, no pun intended with stroke, I'm sorry, but that instantly, self-evidently justifiable kind of territory, right? Because you know that you're demonstrably improving clinical care, quality of life, and the economics of it makes sense. And every indication that we go after is certainly not going to fall into that. Some things are more difficult, right? If it's more of a quality of life impact, what is it worth to give somebody the use of their knee back, right, or avoid joint replacement or something like that? You know, this is where I think the limitations on quality analysis or other attempts to kind of quantify that, they really fall down. Because if you ask anybody that had to go through something like that, whether it's a hip replacement or knee replacement, whatever it might be, they'll say they place a lot more value on it than whatever the classical pharmacoeconomic metrics might be. But the only way we're actually going to get to that point is if we're willing to do the hard work. And I would say, I agree with Eduardo, it's necessary, it's not sufficient. In some cases it can be enabling, depending on what the actual picture looks like. But in most instances, even if they tell you you don't need to do it, I would argue you really better do it.
The other argument you can use is like these companies that will lose the patient in 2-3 years also will inherit patients from other companies who already had these procedures. Yeah. And you're going to incur less costs. So you guys think amongst yourself, it's better for all of you collectively.
Yeah, I personally I've never bought that argument where they say, well, we're only going to have an average client for 3 years and therefore we're only going to focus on that 3-year impact. If you show them at some point they're either paying for the person in part 3 years from now. I mean, think about it, right? If somebody stays with them for 3 years and then they rotate out into another system and somebody rotates out of that system into their system 3 years from now, well, they're paying the fully burdened cost one way or the other. I mean, it may be from 2 or 3 different people, but the reality of it is it's still there. So that's, to me, there's a logic flaw there. Maybe somebody out there is smart enough to explain to me how that math works, but I personally
like in many conditions also, the cost of care for the next 2-3 years is still high enough to justify this. I mean, things like MS or, you know, disabling diseases.
Right.
Question.
Addressing the risk for, say, early stage seems like there's an alternative approach where that end stage or that efficacy stage of phase 2, if you took a strategy where you minimized all that investment for large-scale manufacturing and tried to get a sense of whether it was going to work, would it be, I'd kind of like to have you address that potential risk, because basically you could redo the phase 1 and phase 2 with your new potential cell that's manufactured in a totally different way with maybe not exactly the same profile.
Fair point. You'll have to run bridging studies or other studies. Okay, here's the math on that, at least the example that I like to use. One of the things that I've always tried to impress on people at the company is that every day has value, right? So let's think about it in just purely financial terms if you're a biotech or a pharmaceutical company and you're trying to develop a therapy. Okay, if you develop a new therapy now and it generates $365 million a year in revenue, okay, most people, I think, we would recognize, for a small company, hey, that's hallelujah, right? But for bigger companies, they look at that, it doesn't even register with them, right? But so let's multiply that number by 2 or 3. But for simplicity, let's just start with the 365, okay? That means that every day you are delayed in terms of getting to market is worth $1 million in the future. And so if it takes you a year longer, or 2 years longer, or 2.5 or 3 years longer. And here's the other thing that's fighting against you, and this piece of it doesn't make any sense to me, but it's the reality that we live in. Our IP is going to have a finite lifespan. So if you take a year longer, it gets shorter, the effective lifespan. If it takes 2 years longer, it gets even shorter, right? Well, if you're an investor, you can do the discounted, the NPV on that analysis and say, what's the trade-off there? So I would say start with that analysis and ask yourself, how much longer is it going to take me to get to where I want to go? And what's the financial penalty that I ultimately may pay? And that's going to have a lot to do with the size of the indication, the size of the market opportunity, and everything else. It's not an illogical way to think about it if you're capital constrained at the outset. But you're going to be paying a price for it, right? There's no such thing as a free lunch. So you're either trading value later, or you're going to have to make that investment sooner. And you need to think about the all-in cost of that.
Do you think it could get more therapies into the initial stages?
I'm not sure what that.
I think that what you're suggesting is what most people are doing. So
out of capital constraint.
I think that's the current situation today.
Yeah.
And I think that there's a way in between. And I think that Gil, you mentioned it, which is you can do, let's face it, what we call here phase 1. It's usually a very early phase 2A. So usually we do it with patients and we have a sense of whether it works or not. And I think that that's the time at which you really should commit the funds to do the developmental work at the same time as you do your phase 2 clinical trials. But if you wait until you finish your phase 2 and just before starting phase 3 you redesign your whole process again, well, I think that this is it's a bad decision. I think that then you didn't believe that the company was going to make it, and then you shouldn't be investing in the first place.
And it's not a serial process, right? As you're going through phase 1, phase 2, you can, in the background, begin some development work with that idea of getting to commercial manufacturing. And certainly we understand that you can't commit a lot of resources to that development at that stage, but certainly anything you do early on with the focus of the eventual large-scale commercial manufacturing will certainly be helpful.
Yeah, I mean, one way to do it is you don't build it, you rent it, right? You work with the people that have already invested the enormous amounts of money in the infrastructure, and you say, you got to help us get to a certain point, right? And then we know we can, with high likelihood, get to the finish line from there. And I think that's how you solve that problem.
But the other thing is, I would add, there's things that you should be doing in phase 1. If you're going to invest your dollars, lock down your media, because that's one of the things that will change. If you change your media in phase 3 or phase 2, that will get you. So it makes it very difficult to prove comparability. So you may be able to keep your scale, some of your hardware small, but there's certain things you should be pulling out that are going to affect the characterization of your cells. So it's kind of pick and choose. You can pick and choose your battles at that point. Sorry guys, I'd like to thank the organizers for letting us have this session. We're all going to be around if you have additional questions, and I want to thank the panel again for all your help.